I'm thrilled to be able to welcome our two speakers today who come to us from the Ontario Hereditary Cancer Research Network. And it's going to be a little bit of a different talk and really to generate some interest in getting involved in research and the impact that that research can have on individuals like yourself. So I'll just do a quick introduction of our speakers today, because we're thrilled to have Dr. Raymond Kim and Lauren Hughes with us here today. Dr. Kim is a clinician scientist with a large clinical practice specializing in hereditary cancer syndromes. He's the medical director of the Familial Cancer Clinic and Cancer Early Detection Program at Princess Margaret's Cancer Centre here in Toronto. He is the leader of the Ontario Hereditary Cancer Research Network, head of the Provincial Genetics Program at Ontario Health, and chair of the CCMG Canada-wide Cancer Genetics and Genomics Community of Practice. So he wears a lot of hats. He's really important in bringing the research to clinical practice and ensuring that patients are the ones that benefit from the research that we're generating. So it'll be very interesting to hear some of his perspective today. And I'd also like to introduce Lauren Hughes, who's a genetic counselor and the program manager of the Ontario Hereditary Cancer Research Network at the Ontario Institute for Cancer Research. She received her MS degree in genetic counseling from the University of Michigan, and she has over a decade of experience in clinical genetics. So again, really important perspectives that are both of our guests will bring to us today. So I'm going to put myself on mute, and I'll just remind everyone that as we're going through the presentation and as you have questions or comments, a discussion point, please put it in the Q&A at the bottom. And Dr. Katsopoulos and I will be watching those questions, and we'll be able to pose your questions to our speakers today and have a discussion at the end of the formal presentation. So please keep your questions coming as we go through these presentations, and we'll look forward to having that discussion when we're finished the presentation. So thank you, Dr. Kim and Lauren, and I'm going to hand it over to you.
Great. Thanks, Kelly, Joanne, and the Women's College team for inviting us to share some of the work we are doing at the Ontario Hereditary Cancer Research Network, and particularly all the support the Women's College group has done so far. And really, we learned a lot of lessons from the great work that is done, led by the team here at Women's College. And we wanted to expand it provincially and for all hereditary cancer patients. So I'm Raymond Kim, and Lauren Hughes is my partner in crime. So next slide, Lauren. So the objectives today, I know that this is a mixed audience. It includes patients, providers, and members of the public, is to give you an update on cancer genetics in Ontario, who else is out there, and what remaining questions and gaps result from that, what we are doing at the Ontario Hereditary Cancer Research Network, and what's next, and its impact on clinical cancer genetics research. Next slide. So I like to put up this slide. Many of you will know this. I am talking about hereditary cancer patients, and I have advocated for that these patients are Ontario's golden opportunity in solving the cancer puzzle. Hereditary cancer accounts for approximately 10% of all cancers. They have a unique life cycle with various checkpoints of opportunity. As you watch these patients' journeys, you can see that they are born with this genetic change. We colloquially call them previvors in the lay press. They're otherwise very healthy, right? They don't have cancer, but because they're born with that genetic change, they undergo intensive surveillance, often with imaging of the breast. Some organs don't get any imaging, such as the ovaries. Or if you have a mutation in a different gene, you may have endoscopy or images of your brain and spine. Hopefully, the surveillance detects the cancer early. Many patients, unfortunately, do develop cancer. They undergo some precision cancer therapy based on their genetic change, ultimately, hopefully survive that cancer and have the imaging resume at the following checkpoint. Many times they undergo a second cancer, whether it be in the same organ or a different organ in their body. And it's because that genetic change is present in their entire body. Now, over 100 genes with different types of genotype-specific surveillance are currently known, and there are receptor precision cancer therapies that are based on that specific genetic change. However, due to this life cycle and the various checkpoints, we believe that these families are very valuable in informing us in cancer early detection, prevention, precision therapy, and gene discovery. Next slide. So after much advocacy and lobbying and reviewing, genetic testing in Ontario has had a rebirth. For those who know that BRCA1 and 2 genetic testing occurred at the turn of the century, which was approximately over 20 years ago. And at that time, bits and pieces of the BRCA gene and BRCA1 and 2 genes were being sequenced in different areas of the province. And then 10 to 15 years later, gene panel testing became on vogue, and that was being sent out of country because most of the academic labs and hospital labs in Ontario were not sequencing those genes. And then Cancer Care Ontario stepped in and reviewed the current state and met with many stakeholders to come up with a gene list that would be available for Ontario patients and be done in Ontario labs. And this is just a snapshot of the 76 genes that are available in the 8 provincially funded labs as of April 1st. It's not an April Fool's joke, but it actually happened. And the testing criteria has changed to be more liberal and less restrictive, hopefully allowing people to have access to these genetic changes and genetic testing. And the 19 genes that are covered in a hereditary breast and ovarian cancer panel are encompassed here, and importantly include prostate cancer as an indication for patients to have genetic testing. The usual suspects are here, such as BRCA1, BRCA2. A gene of interest for the group here would be PALB2, partner and localizer of BRCA2, which is an emerging cancer risk gene approaching that of the BRCA1 and 2. So equally as important, but not as common. And other genetic mutations are detectable in the genes listed here, such as P53, CDH1, etc. So next slide, please. So specifically for hereditary breast and ovarian cancer, these genetic testing guidelines are updated initially every 6 months and probably annually to make sure we've got the right gene panel and sure we have the right criteria. They are listed here. Some of the changes that occurred over this iteration include that it used to be only breast cancer patients under 35 would get genetic testing. Now it is in fact increased to breast cancer patients under 45, irrespective of your family history. In particular, triple negative breast cancer associated with BRCA is also highlighted as a criteria. Invasive epithelial ovarian cancer, and also peritoneal cancer. Prostate cancer is on the list too, and pancreatic cancer is on the list too. Previously, they were not even eligible for in-province genetic testing for the lab. Next slide, please. So I wanted to take a brief moment and talk about personalized medicine as they relate to patients who have an inherited genetic change in many of the different genes that we had talked about in the previous slide. So in a BRCA patient, the pathway that is affected in these patients is called homologous recombination. I am a geneticist, and it's interesting that the gene mutations involved in hereditary cancer are involved in keeping the integrity of the genetic material in the body pristine. So in homologous recombination, the body is consistently having DNA damages from various sorts of sources. And they have various types of mechanisms to fix that. Homologous recombination is one of them. It uses the double strand of DNA. There is a sense strand and an antisense strand. And the Mother Nature had engineered it this way. So when damage occurs, you could use a homologous strand to try to repair some DNA damage. And this is where BRCA1 and BRCA2 actually works. If there is a mistake, it looks at the homologous chromosome and then tries to repair it. If you're not able to repair it, though, you can undergo another mechanism. It's called base excision repair. And why am I talking about base excision repair in the context of homologous recombination? It's in fact that a drug targeting that pathway actually allows patients to have a specific treatment. So by targeting a backup pathway that these patients who have a BRCA deficiency or BRCA problem try to use, if you block that pathway too, and those patients can't use that pathway to repair the DNA, that will cause the cell to die, which is what we want in cancer cells. So homologous recombination cells have a problem with repairing their DNA. They try to repair it using base excision repair, which is what a PARP inhibitor also affects. And then the cell can't survive. So basically, that's a complicated scientific way to say that because we know these genetic changes in these families, we're able to look inside the cell and try to figure out what drugs are going to target those specific pathways. And that's a big success in the BRCA space. Similarly, in the BRCA1 carriers particularly, there's another pathway of growth that these patients use in their breast tissue specifically. And it's called the NF-kappa B pathway or rank ligand pathway. And interestingly, that biology is mirrored in osteoporosis. And there's actually an osteoporosis drug that also regulates this pathway. So serendipitously, the researchers have found that using this osteoporosis drug may prevent cancer in these patients, and that's denosumab. And the Women's College Group has extensive experience in looking at this and is potentially a potential drug that can prevent or at least delay cancer, particularly in the breast. There is another ongoing clinical trial by the CCTG, Canadian Cancer Trials Group, that is looking at the role of aspirin in decreasing the likelihood of ovarian cancer in BRCA carriers. And the idea is that aspirin prevents inflammation, and that inflammation leads to a lot of cell growth, leading to cancer. So there's an ongoing clinical trial called OV25, where the investigators here are looking for patients who are BRCA carriers and administering aspirin to see if it decreases the ovarian cancer risk. I wanted to touch a little bit on something that's actually emerging out of the mRNA vaccination literature. I don't have to tell everybody the importance of mRNA vaccines as it was related to COVID. But mRNA vaccines, one of the interesting things about them is that we are able to engineer different types of them very quickly. So the mRNA vaccination community is looking at, well, maybe we can come up with a cancer vaccine. There are vaccines known that prevent cancer, particularly in, such as HPV vaccination, hepatitis vaccination as it relates to liver cancer. So vaccinating patients does have a role in cancer prevention. And particularly in the hereditary cancer patients, they are using those well-known cellular pathways or scientific discoveries to really engineer vaccines, because of the mRNA technology, allows them to be very nimble. And we know that there are a few ongoing trials of BRCA patients receiving mRNA vaccinations and Lynch syndrome patients who have hereditary colon cancer to receive vaccines to see if it delays cancer onset. And a lot of them use the immune system to boost that immune system and the cancer surveillance the immune system has to clear out cancer cells. Next slide, please. So I wanted to talk a little bit about cancer genetics today for the patient who lives in Ontario. Ontario is a big province geographically, and clinically, patients are seen in various different clinics. Despite us having a publicly funded healthcare system where we now have genetic testing that is uniform, 76 genes across the different labs, irrespective of where we live, carriers still face some barriers. They see different genetics clinicians, they see different family doctors, and there's no unified registry or network for Ontario hereditary cancer syndrome carriers to interact with each other on a clinical or scientific basis. So there's lack of standardization of surveillance guidelines. There is inequitable access to research and expertise. And for us in the genetics community, we see that familial cascade testing is underutilized, meaning that that's a lost opportunity. Because genetic testing for a cancer patient, yes, it's great for them, but the real opportunity for us in the genetics community is to identify those other family members at risk and intervene very early where we can actually prevent cancer from actually happening. Next slide, please. So this is equally important is to figure out the perspective of the patient. So Lauren is going to explore and expand on some of the questions, some of the answers that we got from our patient partners at the Ontario Hereditary Cancer Research Network. And when we asked them, what would be helpful for you or your family member in inherited cancer genetic variant that increased your chance of developing cancer? So it's a very charged question, is how can we help you? So we will be going over that in the second half of the slide. Next slide, please. So this disparity and conundrum and barriers can be best illustrated with a case example of a newly identified BRCA1 carrier in a community hospital living in rural Ontario, where high-risk screening and awareness of this fairly well-known genetic condition is limited. They are only able to access highly specialized screening through MRI and mammography, but maybe they don't have an MRI machine that is close to them. Or maybe they don't have a mammogram that is close to them. Maybe they don't even have a family doctor because their family doctor just left. So these are barriers in Ontario that are real. They may have to travel to have that mastectomy, which is recommended in some individuals who have that gene mutation. And they have limited access to research. And carriers have a great thirst for knowledge and enthusiasm to be involved in pushing the boundaries of our knowledge in their condition. They may be involved in a community hospital that it doesn't know where research is available. And there may be limited research agreements between those hospitals to larger academic centers that allow us to enroll these patients. So really a challenge for one of patient A here. Next slide, please. This is contrast to a newly identified PALB2 carrier that's maybe identified in the large research enterprise here at Women's College Hospital. They have access to high-risk screening, many radiologists who have expertise in interpreting subtle changes in MRIs. The right sequences are requested. Follow-up ultrasounds, follow-up mammograms are ordered on a protocolized basis. the support of the genetics clinic and oncologists and the fertility clinics. An entire comprehensive program is available here, and access to research programs and clinical trials within the hospital are available. However, despite this, there is limited data on an aggregate scale across the province on cancer risk, and there are no standardized Ontario screening guidelines and surgical recommendations for all hereditary cancer patients, and particularly for PALB2, where unlike BRCA1 and BRCA2, these guidelines are available. PALB2, it is a evolving target, and the data is always emerging because these carriers are actually much harder to ascertain and reside less frequently when we do the genetic testing. Next slide, please. So the genetics community recognized this, partnered with the Ontario Institute for Cancer Research. Just before COVID hit, we slid under the radar and were able to do a in-person think tank on February 10th of 2020. A lot of familiar faces here participated in that, and we all agreed that the Ontario cancer genetics community should work together to make cancer genetics and the lives of our cancer patients better through development of a program and research network that can enroll patients. Next slide, please. So the Ontario Hereditary Cancer Research is has a vision to use the information from individuals with hereditary cancer syndrome to understand the biology, prevention, and early detection and treatment of all cancer. It's funded by the Ontario Institute for Cancer Research over 5 years and is a $4.5 million initiative. The goals are to identify all confirmed hereditary cancer carriers in Ontario, engage the community on the database development, populate the OHCRN database and develop surveillance guidelines, and enable genomics and gene discovery for these patients that we enroll. So next slide, please. So we will work with current existing initiatives, such as the Familial Breast Cancer Research Unit at Women's College Hospital that has a deep footprint and legacy in the breast cancer research space. However, there are other pockets of research initiatives across the province, such as the Familial GI Cancer Registry, led by Zane Cohen and Steve Gallinger, the Pediatric Cancer Genetics Program, led by David Malkin, the Ontario Familial Breast Cancer Registry at Mount Sinai with Irene Androulis. But there are other genes that are not captured in these research registries, and many patients that otherwise are left orphaned. And we at the Ontario Hereditary Cancer Research wanted to find a mechanism to be gene agnostic, but all encompassing when it comes to hereditary cancer. Next slide, please. So a bit about the Ontario Institute for Cancer Research and Cancer Genetics there. I'm going to turn this over to Lauren Hughes, who's going to explain what the OICR is all about and how the OHCRN fits into this large research enterprise across the street.
Thanks, Dr. Kim. So the Ontario Institute for Cancer Research is really in a unique position to be able to support the development of a cancer genetics research network. For those of you who aren't familiar with OICR, it includes 3 main initiatives: clinical translation, therapeutic innovation, and adaptive oncology, which is the branch that OHCRN fits into. The adaptive oncology program brings together researchers with expertise in genomics, bioinformatics, machine learning, imaging, pathology, among others. So this slide is really just meant to demonstrate the great opportunity we have to work with people and resources across many different disciplines to build something comprehensive. So as Dr. Kim highlighted earlier, this project is very patient-driven. And in order to ensure that what we are building is truly what patients want, we reached out to our patient partners who either have or are at risk of hereditary cancer for their input on what was important to them. And their responses are highlighted here and included things like access to high-risk screening and preventative surgery, access to experts, patient support, accessible testing options for their families, therapeutic intervention, personalized medicine, and enrollment in clinical trials. So these are the features that we're trying to keep top of mind as we build out the program. And one of the current challenges is that some of these cancer syndromes that we're trying to study are quite rare. So in order to achieve improved clinical care in all of these areas, we need a way to bring together as many patients and researchers as many as possible to support the work that actually informs these clinical decisions. So keeping this patient partner feedback in mind, we are building out an online Ontario Genetics Hub. So we'll develop a website with resources for patients and clinicians, including information about clinics, labs, and advocacy groups. We'll be able to link out to provincial health organizations like Ontario Health and Cancer Care Ontario to better connect the cancer genetics community. There will also be a portal into the research registry with different options for enrollment. So for our first step, we're focused on building out registry enrollment by clinicians and by patients. For clinician enrollment, a care provider will be able to obtain consent for contact from their patient. And then for patient enrollment, patients will be able to go online and self-identify as a carrier and provide their own consent for contact by OHCRN. And then more detailed permissions would be obtained later to fully enroll the individual into the registry. The first version of OHCRN will focus on confirmed carriers of pathogenic variants or mutations, but we're building this registry to really be able to encompass everyone who accesses hereditary cancer genetic testing in Ontario. This includes both individuals with and without cancer diagnoses. So in order to build a registry that's beneficial to patients and researchers that also respects patient autonomy means that we must be very thoughtful when it comes to developing the consent. So things that we're keeping in mind again as we build it out are obtaining consent to register into the registry, to release and share clinical and genomic data, to participate in anonymized research, to be recontacted by the program, to connect with existing research registries, and to link to external databases. So what we're actually building is a registry that will centralize patient clinical information, such as their cancer status and type, with their genetic or genomic data, such as their molecular genetic test results. The registry will also link out to external databases that will help capture additional clinical information. For example, a patient's surgical history or maybe their medication history. One of the biggest challenges of this project for us that we're working to overcome is that it requires pulling data from a variety of sources stored in non-standardized formats. All of this information is currently being captured in different ways by different organizations. So when we first started thinking about centralizing this patient information, I will admit it seemed initially relatively simple. We would gather patient clinical and genomic information and consolidate it with any research data that was already being collected. We'd make it available to approved researchers and projects, and this would drive cancer knowledge forward. This is still very much a high-level overview of what we are doing, but the collection of data has become more complex over time as we dig deeper. You start to see the complexity become apparent as we consider the multitude of resources that can contribute to these different information categories. For example, patient clinical data can come from genetics clinics, can come from administrative health databases, other existing registries, and so forth. And then as we dig further, we're discovering that even those secondary contributors have resources that need to be considered. So what initially seemed like a straightforward data collection is actually becoming quite a bit more complicated. So as we get into the weeds of data collection and consolidation, I think it's a good time to take a step back and ask the question, do patients and clinicians even want registries? Is all of this work to bring data together worth it? I mean, obviously we think it is, but what do patients have to say about it? So this paper from last year looked at addressing that exact question. ICON is a research database in Australia for adult carriers of germline pathogenic variants in cancer predisposition genes. And in this database, patient anonymized data is available for research. And so anonymized data is very important in helping inform broad clinical care and health policy. but Australia does not yet have an active national registry with re-identifiable information. And this is the key with the registry, is that storing identifiable and linkable information is necessary to be able to facilitate patient enrollment in things like clinical trials and to be able to connect to additional health data. So this ICON group interviewed individuals who had received care from a familial cancer clinic in Australia and asked for feedback about the establishment of a national genetic and genomics registry. So the short answer to the question is yes, patients and clinicians do want registries and to participate in them, but they had many thoughts that anyone building a research registry, including us at OHCRN, must consider. So these include making privacy and consent a priority, controlling access to data, ensuring protection from discrimination, ensuring that the registry collaborates with other existing research initiatives. The interviewees also commented on the positive impact of registries, such as the opportunity to standardize and connect with experts, opportunity for gene discovery and variant reclassification, and the potential for mutual benefit. And this was a theme, is that it really needs to be two-way communication where patients can provide their information and consent and contribute to research, but also receive up-to-date information and communication back from researchers. Many of the interviewees talked about the incentive to improve their own care and help their families. And I thought these two quotes on the right were particularly indicative of their motivation. So the more information we can supply, I think the better it's going to be for future generations. And if there are treatments or programs or something you can do to try to help, then yes, it's good to know those are out there. So generally, people are very positive and interested in sharing their genetic information for the personal benefit as well as the greater good. The idea of a genetics registry is not entirely new. We've talked about the existing registries in Ontario that function generally separately. And there are other groups worldwide also working on developing more comprehensive research registries. And the ones listed here are included because they're built within similar healthcare systems to Canada. Many of them are also siloed and function in an anonymized fashion, similar to the ICON database that I mentioned previously. The UK especially has several efforts to try and consolidate clinical and genomic data, but even with their national health system, there are limitations to the linkage and use of identified information. And then ERN Genturis is a series of systems of healthcare in Europe, and they developed it to try and facilitate sharing of medical knowledge and expertise across their borders. And as we build out OHCRN, we do continue to explore these related projects, because for many rare cancer syndromes, the only way to truly profile them will be to connect with as many people and groups as possible. So to give you an idea of some of the use cases or questions that we're hoping to answer, centralization of patient information and building of a larger data repository will allow researchers to address some of the questions listed here. So for example, how many BRCA1 carriers are there in Ontario? What are the cancer rates of BRCA2 carriers in Ontario? What's the incidence of ovarian cancer and PALB2 carriers? And then thinking about questions more from a patient perspective that we can help answer. How many people in Ontario have the same mutation as me? At what age do most people with my mutation have risk-reducing surgery in Ontario? I'm a BRCA1 carrier. Do I have a higher risk of colon cancer? Or how many people in Ontario with my mutation have had a mastectomy? Is my variant of uncertain significance now significant? I feel like that's a question in clinical care we get quite often. The genetic results we received aren't always as clear-cut as we'd like them to be. And what research studies can I participate in? So to have really a centralized place where both patients and clinicians can go to find out where, how patients can contribute and how clinicians can get involved. So to return back to the patients that Dr. Kim introduced. So thinking forward to when OHCRN is fully up and running, we could really see the benefits of building this network and connections. So if we return to patient A in rural Ontario, who's the carrier of a BRCA1 mutation, they can now register with OHCRN, connect with current research, access specialists, and know when new screening technologies become available. And then patient B in the city center, who's a carrier of a PALB2 mutation, is now better able to connect with current research as well. Their surveillance guidelines and documents that their clinicians are using to make clinical decisions will be based off more evidence and more data that was generated within Ontario. So hopefully the potential impact of OHCRN was clear, and the collaboration and the importance of collaboration in the hereditary cancer space is evidence. The community building that's happening will help eliminate those geographical and institutional barriers and encourage data sharing and collaboration. The final registry will build on existing systems, including imaging across the province, tumor genomics, and health data. And clinical care will benefit from increased cohesiveness, and the connection to research will facilitate clinical trials and genomic discovery. When I was working in clinical care, patients would often ask me how they could help. As Dr. Kim mentioned, carriers are usually very proactive and engaged, and often wanted to know how they could help further scientific discovery, how they can learn more about their specific mutation, how they can build knowledge for their families, etc. Oops, didn't mean to go forward there, apologies. Patients play such an important role in research, and we'd encourage you, those of you who are on the call, to participate where you can, including attending webinars like this. But once OHCRN is up and running, we encourage you to enroll. And then until then, it's important to share the information with your families about your family history, about the inherited risk factors in your family, about research and clinical care. And lastly, another way to stay involved is to volunteer as a patient partner with your clinic, with your hospital, or other organization. Patient voices really help shape research and ensure that progress continues in a responsible and meaningful direction. And with that, we'll end the presentation and turn it back over to the hosts.
Sorry, I have to click all the buttons there, Lauren, to get back on. So thank you so much to Lauren and Dr. Kim. I think it's a really exciting endeavor, and we will be in a position to answer many more questions that are very relevant to so many of the patients that we see in our clinics.
So thank you so much for that. And sorry, I didn't mean to cut you off.
No, go ahead, Joe. You want to start with the questions?
So yeah, so we have a few questions, and Lauren especially actually started speaking to this. And one of our first questions is, what advice to give carriers of variants of unknown significance? So for example, this one individual was tested for a known RAD51C variant, but was instead found to have a BRIP1 variant. And so I know that's a very specific question. So maybe you can just kind of talk, or either you or Dr. Kim or both of you can talk a little bit about what to do if you're a carrier of a VUS.
Let me just take a moment to read this. What advice would you give to carriers of variants that was tested?
I think it's not meant to be so individualized, just maybe just what individuals should be doing if they're known and haven't received much guidance yet.
Yeah, so variants of uncertain significance in the hereditary cancer space, by and large, most of them are not medically actionable. We do not make medical decisions such as you should have intense imaging or have your ovaries removed. So they are this kind of gray result. And to give you a little bit of information on how genetic changes are categorized, they're generally in 3 areas, and there's basically the laboratory uses a scoring system that all the laboratories have agreed to use. And that scoring system is based upon research on patients who have these genetic changes. So most people who have the gene are known to have a pathogenic variant or a likely pathogenic variant. It's just a classification on a probability that it actually causes disease. It's about 90% or 95% certain it causes that disease or more. Benign or likely benign variants are these ones that are genetic changes that don't really affect the gene's function at all. And then you have these variants of uncertain significance in these two genes or in any gene. And those are these gray results. Some type of genetic change, I don't know what it means. It's not enough data in the laboratory to call it benign or pathogenic. So what am I supposed to do with this? It can be unsettling. We understand that. And in most cases, what we do for these patients is the lab just needs more data to kind of up call it to pathogenic or to down call it to benign. So we usually tell these patients to come to the genetics clinic every 1 to 2 years. Depending on your family history and how that tracks in the family, we may say even more frequent. There are some variants of significance that trend towards pathogenic and that are trending towards benign. And sometimes we can get that information from where the family history looks like. I hope that answers your question.
I think you said in 2 important take-home messages to, check in with your genetics team periodically if you are a carrier of a VUS. And you also made an important point that a lot of these are not actionable right now. So there really are no recommendations on how to screen or help prevent disease in these individuals. Thank you, Dr. Kim.
I think at the beginning of the presentation, you did a great job of summarizing where we are now in 2022 in terms of the availability to genetic testing, the access, how guidelines have changed. And I think it's really important to note too, that it really has been much of the research that's gone on internationally that have allowed us to reflect on who we need to be testing to ensure we have the greatest capture of people who are at high risk of cancer. So there is a question from one of our participants today asking if she had genetic testing done 11 years ago, should she have it done again?
I'll let Lauren take that one.
The short answer is yes. As Dr. Kim mentioned, the testing has changed pretty significantly. And even just when I think about when I started in the clinic, we were testing for BRCA1 and 2, and it would take 6 months to get results back. And even a few years before that, they used to send samples to all the different labs across the province to pull together one report. And now we're doing up to a 76 gene panel in Ontario. So the long answer, that's the long answer to the question, but it's changed pretty significantly. So absolutely, we'd recommend checking back in, getting a new referral to your local genetics clinic, whether that's through your family doctor or oncologist or any of the clinicians that you see on a regular basis. It's worthwhile checking back in because 11 years ago, it would have been a very limited panel that was available at that time.
Great. Thanks, Lauren.
Thank you, Lauren. And this next question may also go to Lauren first. Is the timeline for beginning the registry for the OHCRN that you were just reviewing?
Yeah, that's a great question. And we're working with the genome informatics team to get a more complete roadmap, but we're hoping to begin enrollment within the next couple of years. We're just working on the enrollment pathways and the back end of the registry has to be built really. So that's the rough timeline right now. So stay tuned and feel free to reach out for updates at any time to myself.
Mm-hmm. And definitely this community that we're developing here in our monthly virtual series, we will be able to continue to let the people who are involved know about this opportunity when it does become available. So stay tuned within our group too, and we will continue to feed information as we get it. Okay, I see how, and I think we've had this kind of a question before about when to have genetic testing, but maybe we want to review that in terms of age, Lauren. Or Dr. Kim, I'll let you guys take it from here. So thinking about what age should someone come forward for genetic testing, if we're specifically talking about adult onset cancers, we're not talking about the younger onset cancers.
You go for it, Lauren.
Well, usually when we're meeting with a family to talk about genetic testing, in Ontario, we typically don't recommend testing for adult onset conditions under 18. So I'd say the earliest we would test would be 18 clinically. And then it comes down to weighing the pros and cons for the individual patient and the family. So thinking about what the gene we're testing for in the family, so what the average age of onset and when clinical screening would start for that individual. For BRCA1 mutation, you may begin MRIs as early as, in the early 20s. So obviously when it would impact that particular individual's care. So as young as 18, and then tailor that depending on the gene, the screening availability or surgical recommendations, and then how prepared psychologically that particular individual is to receive that news. I mean, we've had patients who, their daughter is 18 and is in the middle of graduating high school and making decisions about colleges, and it's just the timing wasn't right for that particular test, but knowing that in the next couple of years, it's information that she wanted in order to make some clinical care decisions. So Dr. Kim, do you want to add?
Oh, I actually think that this is a moving needle because genetic testing, we can't, in cancer at least, it's hard to separate adult versus kids because the genetic testing is panel based. So some kids who have pediatric cancer disorders that were traditionally thought, oh, this is a peds thing, I can test for 1 or 2 genes. Or this is kind of confusing, I want to test for all 76 genes. We may be diagnosing some children with BRCA1 or 2 genetic changes that happen that really aren't related to their, or could be related to their pediatric cancer. And then also, there's no minimum age for providing consent. Like there is the concept of a mature minor. So if there is a mature minor who is even below the age of 18, where, the arbitrary, you turn an adult at 18, I'm sure a lot of people would say that that's actually not true with some of their children, is that you could have a mature minor who understands the implications of genetic testing and is 15, 16 years old. And if they check off all the boxes in consent, then we could proceed with that. But those ones are generally dealt with in the pediatric context, where the experts in consenting a child for an adult onset condition have that skill set. So, it's a constantly moving target. So it's a very, very, simple question in the past, where the American Association of Pediatrics said exactly what Lauren said, test when they're 18, when it makes a difference. But that guidance was many, many years ago and has kind of gone, out of fashion because the technology is pushing us in a different direction.
So out of curiosity, in that rare case where you're testing a young child for a pediatric disorder and they are found to carry a BRCA1 or 2 mutation, is that disclosed to them and their family at that young age?
Yes.
It's interesting.
Yeah.
And I think there are many issues that families go through in deciding about testing. And
Yes.
we do have amazing clinical experts who can help you and your family in making these decisions. We have amazing genetic counselors throughout the country. At Women's College Hospital, we have a social worker who's dedicated exclusively to this and helping families with genetic conditions and making decisions about when to test children. She's done a talk in the past for us here on the virtual seminar series, and that is posted on our website. And I think there was a question about looking at the past presentations, and yes, they have all been recorded and they are posted. We always find it challenging to find it, so I'm sure you're finding it challenging to find it. I know Joanne found it yesterday.
You can either go through YouTube, and I don't think there's anywhere I can put this link, but maybe we can include it in the next, or maybe on the actual website. We'll try to do a better job, but they're on our Hereditary Cancer Unit website, like the Women's College Research Institute website, and also probably via YouTube if you were searching. but we're also going to try to improve the accessibility of those videos.
Yeah.
So I think that takes us to the end of our questions. Thank you for really interesting talks to both Lauren and Dr. Kim. We appreciate you taking time in your busy schedules. And so I'm very happy to introduce our seminar series topic for next month, which is going to be Dr. Kelly Metcalf. And I'm trying to share the screen here. So next Friday, May 27th, again at noon, Dr. Kelly Metcalf will be talking about the treatment of hereditary breast cancer, which will likely be a big focus on BRCA1 and 2, and maybe a little bit about PALB2 mutation carriers and some of our research there. So we hope you can join us. So again, thank you to our team and for everyone for joining us today. And we look forward to seeing you next month, and have a great weekend.
Thanks, everyone. Bye-bye.